This conference will now be recorded.
Good day, everyone. Thank you very much for joining us today. We have a presentation today from the Green Soil Group titled The Potential for Bioremediation Principles, which is a case study. Thank you very much for joining us. This is one of our webinars into this webinar. It's one of our webinars on NICOLA, and it's one of the ways in which we're trying to disseminate information within the scientific community. I'm going to start by just asking everyone to mute themselves and make sure that your cameras are off, and make sure that you are on mute. We'll have a very exciting 40 minutes to chat about the bioremediation. Just to start off, I want to introduce NICOLA to you guys. Most of you are already members, but if you're not a member and you don't know what NICOLA is, NICOLA stands for the Network for Industrially Contaminated Land in Africa. One of the objectives of NICOLA when it was formed, it was for knowledge sharing in Africa, which is basically providing an African forum for disseminating information and good practices within the remediation or within the contaminated land industry in Africa. This we do through working groups on relevant topics. We've had a few successful working groups where we have come up with position papers. And again, it's in contaminated land training. We've done quite a few contaminated land training within the group, within the NICOLA group, and also with the authorities in South Africa. And we also have an annual conference that is due to happen we normally have an annual conference, but we couldn't have it this year due to COVID. But we will have a general meeting, which the information about that general meeting has been shared already. The other objective is for it's to allow networking, which is through this one or seminars or webinars like this one that we're having. We also have regular evening networking events that we have in different cities, and also industry forum events. The other objective was inspiring the scientific best practice, which is stimulating and coordinated and interdisciplinary projects on collaborative, problem-oriented research and knowledge transfer to address identified needs. And also to cooperate between industry in mining, academia, regulators, service providers, while linking closely with the fields of contaminant hydrogeology, geochemistry, and waste management. We were also very lucky to be awarded for our contribution to land remediation in Africa by the Department of Environment, Forestry and Fisheries in 2019 in a conference that took place in Kimberley. So basically that is NICOLA in a nutshell. And if you have any questions, there is a steering committee. You can just pop us an email, or you can try find anyone within that steering committee would be able to give you extra information if you need. Just in terms of future plans that we have, as I mentioned earlier, we couldn't have our annual conference this year because of the current situation, but we had planned to have a conference with SASTREAM, which is SASTREAM 2021, which would be co-hosted with the Department of Environment, Forestry and Fisheries. At this point, we're looking at a date between the 28th and the 29th of October 2021. and the venue would be at the Spear Wine Farm in Cape Town. The theme for that conference would be for empowering sustainable land management for the future. So we're inviting you. Please avail yourselves and join us in that conference in 2021. I also just wanted to introduce the speakers for today. The Green Soil Group. We have a very exciting webinar today. We have John Dyke and Michaela de Camelis from the Green Soil Group. Just a quick introduction on John. John Dyke is an R&D manager at Green Soil. He has been active in the field of bioremediation for more than 20 years. We will get hopefully all of that experience during this webinar. He finished his PhD on the degradation of chlorinated compounds under anoxic conditions in 2005. After a scientific career, he had been working for several contractors in the field of R&D. Since 2016, he is head of the R&D department at Green Soil, and a contractor specialized in situ and on-site bioremediation. He has extensive experience in laboratory and field research of bioremediation, potential for conventional and emerging contaminants, with a strong emphasis on microbiology. We will also be joined by Michaela. The webinar will be hosted by the two of them. Which Michaela is, her name is Michaela de Camelis, is a sales and project engineer at Green Soil. She is responsible for international business development, identify new opportunities and develop strategic remediation plans for the application of bioremediation. She is also support to the R&D team carrying out laboratory tests and investigation on emerging contaminants. Prior to joining Green Soil, Michaela worked for 5 years as a researcher at Ghent University in Belgium in the field of geoenvironmental engineering. Yeah, thank you, Michaela, thank you, John, and it's all yours. You can take it from here.
Yeah. I hope you can see my full screen now. Thank you for the introduction and the opportunity to present here and to share with you the potential of bioremediation. Today's presentation aims to describe the different bioremediation pathways and points of attention before focusing on 3 different case studies. But first, let me introduce briefly Green Soil. Green Soil is an international general contractor, and despite in some projects we are able to perform some civil works like demolition, our core business is bioremediation on site and in situ. One of our characteristics is also the innovative aspect, because we are always looking into the potentials of bioremediation on new emerging contaminants, but also we explore new biological techniques. Because of our experience, then we are able to provide also fixed price contracts and to take over the liabilities in some cases. Our main clients are oil and gas and chemical companies, and at the moment we work in about 12 countries on different projects, from laboratory tests to pilot tests or full-scale application. We also have a project in South Africa, as you can see, and we always work with local partners. But now, why we actually promote so much bioremediation? Well, that's mainly because our projects are based on the contaminated mass. So our projects costs are driven by the contaminated mass. And unfortunately, when we start a remediation project, we always have a surprise concerning the contaminated mass. Usually, this is always higher than what it was estimated during the preliminary investigation. And if we use pump and treat techniques or physical chemical techniques, those are really dependent on the contaminated mass, and the cost of the project has a big impact. So increasing the contaminated mass, this project cost can increase. Whereas on the other side, biological techniques can control better the cost of their projects, even if the contaminated mass increase. And why we consider biological techniques a robust technology? Well, because our main employees are actually the microorganisms and bacteria present on site in situ. And what we do is to turn the soil into our bioreactor by stimulating the right conditions. So once we have the right condition in the soil, microorganisms will do the rest, and then it's only a matter of time. That's why we also take risk of taking over the liabilities or risk-based contract, in particular with chlorinated compounds. Biological techniques are also more sustainable if we consider the CO2 footprint compared to biopiles of soil washing, as you can see from the right graph. But now I hand over to John, who will dive more into the biological aspects of bioremediation.
Yeah, thank you, Michaela. So I will start right away with one of the basic principles of bioremediation, and that is the assumption that bacteria are everywhere and that nature selects. So that basically means that if you look at a contaminated sites, the bacteria had a chance to adapt to those specific conditions, and that basically the best bacteria are present there, best suitable for the job, so to say. There are some exceptions, of course. One of them, Michaela will also illustrate in one of the practical examples she will speak about later on. But I would say in general, more than 99.9% of our projects, we don't have to add the bacteria because they're already there. In general, some of the factors are basically limiting biodegradation. I will show them later on, but in general, we talk about like factors like oxygen or a suitable electron donor that is kind of limiting the biodegradation because it's not there, or it was there, but not sufficient, so it's already consumed, and therefore limiting biodegradation and biodegradation potential. So as Michaela already indicated, the base idea of bioremediation is to create optimal conditions. So basically you add a compound, for example, several compounds, and you create the right conditions for the degradation pathway. So basically you stimulate the natural degradation. Finally, I would also like to mention that also high concentrations, or talking about source zone, and in general, no problem to also treat with bioremediation. Michaela? Yeah, thank you. So one of the first things, of course, is to consider which kind of degradation pathway you choose. I mean, if you want to stimulate a degradation pathway, you need to know, of course, which one and what you need to do to basically to optimize the process. So what is required to stimulate the process? A couple of things are important, of course, to kind of select the degradation pathways. One of them is, for example, the type of the contaminant. Also geochemistry, soil composition, and hydrology are, of course, very important to make a decision. You also look at, is degradation already going on? For example, you can look at possible degradation products that are already present, which gives you an idea about what kind of degradation is already going on. But you can, of course, also look to the microorganisms. So, for example, with DNA research, you can basically determine which type of microorganisms are present or even active. What are their numbers? So that gives you also an idea about the potential at the site. Finally, also technology is, of course, important. For example, the depth of the contaminant. If you, for example, think about aeration, there are some limitations, for example, to the depth, or you have to adapt your pressure. risk of clogging. For example, if there's a lot of reduced iron presence, you're thinking about aerobic remediation, there will be iron precipitation, iron oxides, hydroxides being formed. So that's also something to take in account. And of course, also time and cost. If you have more time, generally you have more options. If you think, for example, about anaerobic degradation pathways, generally they take a little bit, they're a little bit slower, so they take more time. So if you have the time, generally you also have a little bit more options. And finally, of course, knowledge about the degradation pathway is very important. You need to know which kind of pathway exists, which microorganisms are involved, or maybe which enzymes, which you can also follow up during the remediation. So you have to know your microorganisms. And one of the things, for example, luckily, there's quite some progress in these kind of technologies that are also available commercially. For example, next generation sequencing, as they call it. You can see an example on the right, which looks a bit like a pie with different parts. The different parts are basically groups of bacteria. And in this way, you can have kind of an image of the microbial community that is present in, for example, a well or a soil sample. And it gives you also an idea. For example, in this case, you see it's in the left bottom, you see a big group of bacteria. In this case, these are Dealococcoides, and they make up about 25% of the total community. So that already gives you an idea about the potential and what's going on during your bioremediation. Another one is, it's kind of a rule of thumb. If we talk, for example, about chlorinated compounds, also by looking at the compound itself, basically the amount of chlorine atoms attached to the molecule, you can have an idea which type of degradation pathway is most likely to occur. And that's basically just chemistry. So basically the amount of energy, for example, a bacteria can gain from the transformation of the compounds. So here on the X-axis, you can see the degree of chlorination. So we have on the left side, we have a low chlorinated compound, in this case monochlorobenzene. And on the right side, we have a molecule with a high degree of chlorination, in this case hexachlorobenzene. So what you can see basically on the y-axis is the reactivity, so kind of how likely it is that the process occurs. So in this case, we have oxidative dechlorination, so basically oxidation of the compound, and you can see that it's more likely to happen with low chlorinated compounds. Well, if you look at the highly chlorinated compounds, it's more likely to be reduced. So that's kind of what you can see in this figure. So if we talk about aerobic remediation, or bioremediation, basically, well, the green thing should represent the bacteria. So if you look at the process itself, the contaminant, in this case with hydrocarbons as contaminants, is basically the food of the bacteria. It's what we call the electron donor. So in most cases, that's also the energy and carbon source of the bacteria. So it's basically its food, which is being oxidized to water and carbon dioxide. And then we also need an electron acceptor. In the case of an aerobic degradation pathway, that's oxygen. So it's the same electron acceptor that we humans also use, quite common, the oxygen. But we see later on that there are also some other forms of electron acceptors. And then we also have some nutrients, especially like nitrogen and phosphorus, depending if they are present on the location. But in most cases, you also have to add them so the bacteria can basically multiply and create biomass. So here you can see a little bit more about the characteristics for some of the compounds that we treat with aerobic bioremediation. I already mentioned hydrocarbons. You can also think of low chlorinated compounds. Also compounds like alcohols, ethers, phthalates. But also sometimes we look at some parameters like, for example, COD or TUC, in case you have a complex mixture of different contaminants, so you don't know all the contaminants. If you talk about hydrocarbons in general, it can occur, of course, as an L-nipple, so floating on top of the water, as you can see in the picture. You can have a smear zone by the fluctuation of your water level that the contaminant is being smeared. In general, hydrocarbons have a low solubility and relatively high retardation, so they're not very mobile. So you often also you don't see really big plumes. Of course, there are some exceptions for the more mobile one, like for example the BTEX, the aromatics, for example benzene. It's a bit a different story, of course, but the typical mineral oils don't have that. And as already mentioned, you need oxygen. In general, that's about 2 grams of oxygen per gram of oil. You can see that in the reaction below. It's basically, as I mentioned, the hydrocarbon is being used as food. So part of the hydrocarbons are being used also to create biomass as a kind of the carbon source for the cells. And it's about 40% in the case of aerobic degradation of hydrocarbons. And then about 60% is basically oxidized to gain energy from the process. If you then switch to the anaerobic bioremediation, so kind of similar picture, but here it's the other way around. So the contamination, as you can see, is the electron acceptor basically. So now it's on the right side. In this case, you can see, for example, tetrachloroethene, PCE, trichloroethene, TCE. So the chlorinated ethenes, those are the electron acceptor and therefore being reduced to eventually ethene. In some cases also ethane, depending on the geochemistry. So in this case, basically we have to add an electron donor. So that's basically the food of the bacteria that's often limiting. So in our case, we for example use GLOGS. Of course, you have a lot of different commercial available electron donors. You can think of lactate, vegetable oils. There are a lot of options, of course, but basically you need to add a suitable electron donor to stimulate the process. And some of the characteristics, of course, you have different chlorinated compounds that you can treat with the anaerobic bioremediation or anaerobic reductive dechlorination, as we often call it. You have the chlorinated ethenes. You also have the, for example, hexachlorocyclohexane, chlorinated benzenes, 1,2-dichloroethane. So there are a lot of different ones we can treat. Those are basically fairly water soluble and therefore also quite mobile. So you often see a relatively large plume. And they form, in contrary to the hydrocarbons, they form D-nipple, so it's more dense than water. So we can see that on the picture on the right, basically it sinks. So you often encounter, of course, these contaminants at greater depth, which also kind of determines the design of your system. And as I mentioned, it requires the addition of an electron donor, and basically the function of the electron donor is twofold. So on one hand, it serves, as I just showed, as an electron donor for the process of dechlorination itself. However, you also have to create the right redox conditions. If you talk, for example, the Dealococcoides I mentioned before, this specific group of bacteria is mainly involved in the last two steps of dechlorinating of the chlorinated ethenes. So in this case, the last two steps, as you can see in the picture, from cis-DCE to phenyl chloride and to ethene. And those bacteria are quite picky, so to say. They really like strongly reducing conditions. So therefore, if there, for example, the groundwater contains a lot of nitrate, for example, you first need to reduce the nitrate before those bacteria start to become really active. So that's the other function of your electron donor, that you have to create the right redox conditions. So some of these success factors, at least in our experience, Michaela will go a little bit more into detail about the system we use, but it's basically a continuous circulation of groundwater, which is a bit different than, for example, injection of your substrate or your additions. But it enables us also to follow up the process quite intensively if we want. So the monitoring is a really crucial part of the remediation. So basically you follow up the concentrations of your contaminants, but also, for example, the nutrients level or the electron donor concentration, and also the geochemistry. And it's kind of course, enables you to see if the conditions are okay, or if they are still okay, and if the process is going on. We also, as I already briefly mentioned, we have like additional innovative monitoring techniques to kind of gain like extra evidence that, for example, biological degradation is going on. Caleb will also show this for some of the projects, but if you have, for example, a source zone, then it's not always clear just based on the concentration what's going on. So you can use some additional monitoring techniques to get extra evidence. So if you, for example, a compound-specific stable isotope analysis, which kind of you can use to kind of have extra evidence for biological degradation, and also more the molecular analysis, like for example, quantitative PCR and next generation sequencing, as already explained. And by this, you can basically continuously optimize the process of bioremediation. So then the final slide of my part, some more the theory, some points of attention when you consider bioremediation or you're looking at the site, which factors are really important to take in consideration. Already mentioned the geochemistry, but some of these important factors are, of course, like pH, temperature, electrical conductivity. Bacteria always have a specific range for these factors where they are most active. For example, pH, well, in general, most bacteria are most active at more neutral pH, so the range of 6 to 8. Of course, there are luckily enough exceptions, but this is more like a general guideline. Also, parameters like, for example, iron, COD, nutrients are important. Iron already mentioned a little bit. If you have a lot of reduced iron and you're going to add oxygen, of course you get iron oxidation, so it might clog your wells. pH typically will decrease, so that's also something to take in account. COD, so the chemical oxygen demand, basically kind of indicates how much oxygen you need. So if there are a lot of reducing components are present, can require a lot of oxygen. So just besides the contaminants being oxidized, you can have like sulfides, for example, a lot of reducing compounds or maybe unidentified contaminants that also require a lot of oxygen. So that will also determine the design you need to have for your remediation. In general, I would say the analytical methods. Analytical methods can also be very important. We noticed that we had a project in South America where we basically had a bioremediation of hydrocarbons. So it was only about at the end of the remediation, we kind of saw a stall in the concentration of hydrocarbons, which was a kind of strange because the conditions were optimal. So we couldn't really figure it out what was going on there. So in the end, we decided to ship some samples to that time it was the Netherlands, to have some further research about the material. And then we discovered that the concentration of hydrocarbons analyzed by the lab we use here was much lower than the concentrations that were basically reported by the local lab in Brazil in this case. And then finally we found out that they use a different method where they didn't have a standard pre-treatment. It's called like a fluorosil cleanup, where you basically remove the humic acids. Well, here in Europe, it's quite standard to include that pre-treatment to remove the humic acids, but there it was not done. So basically, we were looking at humic acids instead of hydrocarbons. Of course, that's one mistake you never. Can you go back, Michaela, for the other two? So you don't want to make that mistake, of course. Some extra points. So if you look, for example, if you apply a bioreactor, you have to take in account that bacteria don't really like big changes. So that's something to take in account. But on the other hand, you don't You shouldn't underestimate the power of bacteria and the flexibility. It just takes some extra time. So I would say, and end this with, if you want to really test the full potential of a location, it's best to start with a laboratory or a field test to investigate these. Thank you.
Thanks, John. Now we move from the theoretical aspects of bioremediation to 3 case studies. We'll go through 2 aerobic projects, an in situ and on-site bioremediation projects. And at the end, I will show you an in situ anaerobic project. Doesn't go further. Okay, let's start with a pilot in situ MTBE degradation. This is a pilot test we ran in Belgium, where we had a cocktail of contaminants, but mainly MTBE, high concentration of MTBE were present. We know that conventional bioreactor reach an efficiency between 40 and 60%. That's because of the characteristics of the MTBE, which are very mobile and they don't absorb too much to soil. So conventional treatments don't really work with them sometimes. And biological processes have a potential, but the biodegradation was not always straightforward. The Forfito, which is a Belgian research center, isolated MTBE degrading bacteria from different sites and created a specific consortium of bacteria for the degradation of the MTBE. The project background, well, is a complex industrial location because it's an active site where several contaminants were present. We had MTBE concentration up to 13,000 micrograms a liter. But in addition, we also had high COD and strong reducing conditions. Then because of this site condition, ISCO and multi-phase extraction pilot tests were first performed without success. And because of the actually site condition, bioremediation was never taken into consideration. However, we proposed an in situ bioremediation approach by a groundwater circulation system. So we extract groundwater, we dose nutrients, and we reinfiltrate upstream the groundwater without treating. And in between extraction and infiltration well, we install biosparging lenses to supply air to the soil and to turn the soil into our bioreactor. That's the aim of our circulation system. So to distribute oxygen, nutrients, and bacteria, to mobilize the contaminants, and to transform our soil into our bioreactor. An SBE system was also installed to avoid explosion risk due to high lead levels. And at this site, we also tested the efficiency of the MTBE bioreactor developed by Vito. It's a bioreactor containing the specific consortium of bacteria. And the bioreactor was fed by one extraction well, and once the water was treated, this was reinfiltrated. Here you can see the system layout with the extraction well downstream and infiltration wells upstream. And between, we had all the biosparging lenses. Now, the couple of wells on the top were the ones connected to the phytobioreactor, and you can see the results from the left graph. What I want to highlight from these busy slides is that the time of our pilot test was set to 6 months. And as you can see from the red columns, within 6 months, our contaminants concentration didn't decrease much. So the client was not happy with these results, of course. But we look beyond the concentration levels, what was actually happening in the soil. That's very important when you apply bioremediation, because it's not only, you don't have to look only to the concentration levels, you have to look to the all the parameters in the soil. Well, actually, all the air we were supplying was being used by other processes. iron was being reduced, and we had formation of sulfate. So once all these processes were finished or they reach equilibrium, the ORP increased and we switched from an anaerobic condition to an aerobic actually conditions, environments. The concentration of our contaminants decreased. And luckily, because we looked into the geochemistry, we were able to convince the consultant and the client to prolong the pilots, which at the end was considered successful. Now the graph on the left wants to show the cometabolic degradation of MTBE with the circular line and the BTEX. So once the substrate is gone, you can see it here, the MTBE concentration decrease. And what's the graph on the right? Well, the orange line describes the concentration trend of the monitoring well located close to the phytobioreactor, so close to the point where we were reinfiltrating water coming from the phytobioreactor. And you can see that the concentration in that point of MTB were way lower compared to the other monitoring wells located farther away. And that's because the water passing through the phytobioreactor was bringing with it the consortium of bacteria that then act in situ. So what are the main outcomes of this project? Well, we don't have to look only to the concentration levels. It's possible to change geochemistry conditions in situ to make our bioremediation working. And then it's just a matter of time that the right conditions are there before it actually started, all the microorganisms started creating our bacteria. The phytobioreactor worked with very high efficiency, and we noticed a very positive effect of bioaugmentation. Thanks to the positive results, we are now working on the full scale. So now we move to the aerobic bioremediation on site. This site is also located in Belgium, and the contamination was mainly from xylene in soil and groundwater. What is very important from this project is that we established a risk-based contract, so we had a fixed price, and that's very important because it's directed our decisions during the process of the remediation. So we had 3 contaminated areas in soil and groundwater with about 25,000 cubic meters of contaminated soil. As I mentioned, the contamination was BTEX, but mainly xylene and ethyl benzene. And at the beginning, the idea was to dispose all the soil off site. And because of the high concentration level, it has to be brought to a thermal plant. But our approach was different. We proposed to treat the soil on site. We proposed to treat the 76 tons of contaminated mass until 5 meters, but we also went to 10 meters below ground level and treat the further 7 tons of contaminated mass in groundwater. The approach we proposed was on-site biopiles until 5 meters, lowering the groundwater level, and then an in-situ aerobic remediation until 10 meters by installing biosparging wells. So the way the procedure we used to build biopiles was really important because you can see the buildings surrounding our sites, and there were also houses in the neighborhoods. So it was really important to limit emissions. That's why we built our piles within the excavation area, which limited handling and transport, therefore emissions. So we installed an air extraction system on the bottom of the biopile from which we were extracting contaminated air. And on top of this system, we place our contaminated soil. The biopile was not covered because the aim is to attract actually air from the atmosphere because we want a really aerated soil. And the contaminated air that was being extracted was then treated by, at first, activated carbon. But then we were actually consuming about spending 2,000 euro a week, which was very costly in case of our fixed price contracts. Therefore, we had to look a bit out of the box and look for other alternative techniques to treat the air. And I will show you what our idea was. So we treated the soil in these two areas, and here you can see the location of the groundwater system. And here are the results of the groundwater treatment system, so which work with very high efficiency, even with high concentration levels. We had a stripping tower as polishing step, although the efficiency of the water system was very high. And here the results of the biopiles. Well, this is very like a very nice graph to show, because it's clear that regardless the starting concentration levels of the soil, well, the piles were actually cleaned within 70 days. And we went even further below the objective limits, except one pile that was built under wet conditions. And it's not that we don't like humidity in the biopile. We do like humidity, but when it's too much, it can clog the pores, which should be available for the air. So we rebuilt the pile, and after the pile was built under the right conditions, the soil in 70 days was clean again. But which was the brilliant idea to treat this highly contaminated air? Well, was the idea of our project manager to reuse a clean biopile. So we first clean the biopile, and once the soil reached the objective limits, we use this pile as an air treatment system. That's because the soil was biologically active, containing nutrients and bacteria. So we connected, let's say, biopile with contaminated soil to the BAT, which is our new air treatment system. Very cost efficient because it worked with an efficiency ranging between 70 and 80%. And this allowed us to save up to 90% on activated carbon, which was really,
NICOLA?
Yeah. Hello?
I'm not sure if...
Yeah? Can you hear me?
We're struggling a bit, NICOLA. We're losing you every now and then.
Oops. Hear me?
I can hear Michaela.
Yeah, I'm fine here in KwaZulu-Natal. No problem.
No problem here as well, Michaela.
Oh,
Okay. Can I go?
okay, sorry.
No problem.
It's probably my side.
It's okay. So with this air treatment system, we saved up to 90% of activated carbon just by inverting the air system. Before we were extracting air from the biopile, and now we inject contaminated air through the biopile, and we cover the pile because we don't want contaminated air in the atmosphere. Then once we finish the soil and air treatment, we install the in-situ aerobic remediation in the third area by installing biosparging lenses. And we only use biosparging and not a circulation system because already enough nutrients were present as we analyzed soil and groundwater levels. And we also installed the SVE system. So here you can see the results of the groundwater concentration. In orange, you have the baseline concentration from June, but we actually started the system in January. So within 9 months, we already reached 73% of efficiency. And after 2 years, this efficiency increased further to about 95%. So the conclusion of this project, well, with this project, we wanted to show how it is possible to remediate high contaminated soil, groundwater, and air 100% biologically. So bioremediation can handle also very high concentration levels. In addition, we had no creation of waste and less transport movements because we didn't dispose any soil off sites, and we had to less activated carbon to dispose also going in and out. And also, like, what is very important is that the type of contract in this case, fixed price contracts, drove or stimulated innovation part. Okay, now we move to the last case study. Is an in situ anaerobic remediation project in Spain, where we first performed laboratory and field tests to see if actually anaerobic degradation would work. Is an industrial site in the one of the ports of Spain, which was heavily impacted with chlorinated 18s and chloroform. You can see the high concentration of PCE and vinyl chloride present, but also chloroform was present at very high concentration. And we know from literature that concentration higher than 5,000 micrograms a liter of chloroform can inhibit the growth of the allococcodies, which are the bacteria that are responsible for the degradation of our compounds, and especially for the last part of the degradation pathway, which goes from vinyl chloride to ethenes, which is our harmless product. In addition to this high concentration, we also had challenging conditions on site. The groundwater was brackish and salt, and high concentration of sulfates, and EC was also very high. So despite these challenging conditions, we propose a biological approach by a groundwater circulation system. So we extract groundwater, we dose an electron donor, and we reinject water. And again, this circulation is to distribute the electron donor, bacteria, to mobilize the contaminants, and to stimulate the anaerobic condition in situ, to transform the soil into our bioreactor. In this case, we equip the system with a DNAPL removal system to separate the DNAPL which were extracted from the circulation groundwater. The area, as you can see, was divided into 3 areas. We had the source area and 2 outer areas. That's because we didn't want to extract water from the source with high concentration and to reinject this water in the outer areas where concentration levels were lower. So we installed 3 different systems, independent, and also the wells are flexible because they can work for as both extraction and infiltration wells based on our needs. So the DNAPL removal system extracted in 1 year about 70 liters of DNAPL, but the concentration levels suggested that still DNAPLs were present on site. Now, from this graph, you can see on the right the trend of a monitoring well in the source area, and on the right, trend of a monitoring well in the outer area. Well, the degradation products in the source, in this case, the decrease after the start of the remediation process. Especially 18 increase, reaching concentration up to 26,000 micrograms a liter, which is a really high amount. But this is a really good graph to show. On the other side, other monitoring wells on the source area presented a very fluctuating trend. They will show an example in the next slide. On the contrary, on the outer area, you can see that after the starting of the remediation, the TCE concentration went down and remained low. Therefore, we switched from an active to a passive system. Now here you can see on the right the graph I was mentioning before with fluctuating concentration. So the consultant and the client wanted to understand what's happening because this is not nice to see. And we perform a molecular analysis to see which microorganism responsible to anaerobically degrade our contaminants were present. Well, thanks to this analysis, we were able to demonstrate that bioremediation was actually ongoing because you can see the different microorganisms and enzymes we analyzed. And you can see that from the beginning, so the blue columns, the number, the cell or gene copies a liter increase really of big amounts after the starting of the remediation. Especially, you can see geobacter, they grow really close to the apple and they are able to withstand high concentration levels. So you can see how they increase in number. So we see that the source zone is a bit unclear based on the concentration levels, but you can see that members of the allococcoides, geobacters were all present in high numbers and increased with time. So this was a very good sign. In addition, we also measured the biological degradation rates because we had a limited amount of data at the beginning. We didn't know the exact contaminated mass. So to have an idea of the contaminated mass removed by bioremediation, we estimated these values by degradation rates. Now, the degradation rate based on the literature was measured considering only two groups of bacteria, the allococcoides and geobacters. That we found during our molecular analysis. Therefore, actually, this number is a bit an underestimation because it doesn't take into account all the group of bacteria involved in the anaerobic bioremediation degradation. For this reason, we performed site laboratory tests. The laboratory tests were performed in one test in our laboratory, and another test was performed from Wageningen University in the Netherlands. And laboratory tests are normally run in optimal conditions. And because of this, actually we took the 10% of the actual degradation rates found from Wageningen University, because we work with site condition, not with laboratory condition. So it's just to be conservative, let's say. So we then obtained this PCE mass removed, and we believe that we are in the range of 3,000 to 7,000 kilos of PCE removed since the beginning of the remediation. Now, to conclude, yeah, this project is an example of how bioremediation can work also in very challenging conditions like brackish environments, but also high concentration of PCE combination of chloroform. We demonstrated through molecular analysis, qPCR analysis as well, that the allococcoides were present in high number, which demonstrate actually that bioremediation is ongoing. And thanks to the combination of different monitoring techniques, we could explain the degradation process. We could make the consultant and the client more confident with our work. Because sometimes, especially with bioremediation, we don't have to look always only to the concentration levels, as was the case also for the first case study, but we have to look also to the other parameters which are involved in the bioremediation. Okay, thank you for your attention. And sorry, we went a bit longer than expected. I hope everything was clear. Otherwise, you can see our contacts and feel free to drop us an email with your questions and doubts. Hello?
Linda?
Thank you.
Thank you very
Thank
much,
you.
Michaela.
Thank you, Michaela.
Linda?
Thank you, John. I think before we take questions, I'm just going to share the password for the CPP points. Rochelle, if you can just stop recording.
This conference will now be recorded.
Apologies, Michaela. Do you mind sharing your contact details? Great. Thank you very much.
Yeah, no problem.
Any questions?
We're overloaded of information.
That was very clear, Michaela.
There's a question, can you make a comment about the efficiency of in situ versus ex situ systems?
Well, when we talk about ex situ, do you mean like to dispose of site soil, or like on-site soil treatments? Because in that case, well, when you work in situ, you actually have relative low cost, because you don't have a lot of costs related to transport, excavations, but you can actually work there without disturbing the site operations, because, for instance, we also have projects, bioremediation projects, where we installed in situ system under buildings, under a windmill. So it's still possible with bioremediation to work where in active sites where buildings are present, and this of course saves your cost and handling of the contaminated material.
Yeah, so the question is more around, so the system, the examples you showed, you had kind of like systems that where you had wells and were injecting, versus like your biopile approach where you're kind of excavating the soil. I just wondered if there's a relatively, whether, I guess I assume that if you're able to excavate and do the biopiling, that you're able to get better contact, and like you made the modification in that example, you know, if things are not working, whereas you can't really do that if it's wells in the ground and the materials underneath buildings or that sort of thing.
Of course, like if you are able to excavate and work with biopiles, that can be really cost efficient, but it's not always possible because it requires quite some space. In terms of efficiency, they both techniques can be very high efficient. But then it depends, of course, on site conditions, site soil characteristics, as well as contaminant concentration levels and characteristics.
I can maybe add that typically for the biopiles, or basically excavation, it's done mainly for like the unsaturated zone and just a small part in the saturated zone. So just the first meter, for example, basically if you can get out the smear zone, if you talk about hydrocarbons, that's then you have a high efficiency with the biopile. So for the in situ system, that's typically more for the deeper part of the saturated zone, of course, depending on the specific site conditions.
Yeah.
Thank you, Haley. Thanks, Michaela. Thanks, John. And there's another question. How do you, from Lindsay? How do you find the timeframes vary between aerobic and anaerobic treatment? Of course, concentrations and site conditions are variable, but in general.
I can maybe answer that, Michaela.
Yeah.
Yeah, that's a difficult one. I mean, typically there is you choose either of like either an aerobic or anaerobic approach depending on the contaminant. So in that way, it's not very comparable, but there are of course also some contaminants, for example, 1,2-dichloroethane, which can be degraded both aerobically quite well and also anaerobically quite well. Yeah, it's difficult to say. I mean, in general, that's aerobic degradation can be like 5 to 10 times faster, but that also that's more the degradation process itself. Then you also have to take in account the dosage of your air, basically the efficiency of your aeration system. So it's a difficult one. I would say, yeah, maybe it's more than in the range. If you sum up, it's more like maybe twice as fast, for example, or somewhere between a factor 2 and up to 5, depending on the system, if you have an aerobic remediation.
All right, thanks, John. I think there's another question from Adolf. What are some of the key parameters to consider for in situ groundwater bioremediation?
In general, we look into the geochemistry, so redox. We look into the sulfate iron levels, the nutrients present also. So the what are the key parameters? The soil structure to design the distance between wells to groundwater flow as well. So we look into like several parameters, and so all of them they will give us the full picture for the specific in situ design.
Thanks, Michaela. I hope that answers Adolf. I think another question from Simo. Are you using ethene as a proxy for the efficiency of treatment for both DNAPL and LNAPL plumes?
Can you maybe repeat the last part? Was for me not the ethene for.
Are you using ethene as a proxy for the efficiency of treatment for both DNAPL and LNAPL plumes?
Yeah, ethene in general, if I understand it correctly, otherwise correct me, but ethene we mainly use for, of course, for what Michaela mentioned, for the anaerobic reductive dechlorination. So if you have chlorinated ethenes, that's basically the final product. So then there's, in that case, it's a quite important parameter to follow up. For the aerobic one, yes, just showing one of the sheets, typically the degradation products are basically carbon dioxide and water, so you don't have any really specific degradation product that you can follow up under aerobic conditions. So in that case, you look at different factors, like for example oxygen consumption, these kind of things, or just looking at the microbes itself. So basically count them, so to say, for example, via quantitative PCR.
Thanks, John. I hope that answers Simo. Maybe we have time just for one last question, if there is one. Doesn't look like it. Thank you very much. I think that's it for the day. Thanks, John.
Also,
thanks,
thank you.
Michaela, for the interesting. There's one more question. Can I take it quickly? In project number 1 for MTBE bioreactor, do you need to change frequently the microorganisms inside the reactor, or is it a system that is auto-regulated in terms of biomass growing?
Shall I take that one, Michaela?
Yeah, I think this year.
We don't have to replace it. So basically, that's the phyto bioreactor. That's so just it's inoculated just before the project. So basically, only one time. And they use, at least for the pilot system, they use a kind of special kind of carrier material inside of the bioreactor, basically to retain the bacteria, so they can attach themselves quite quickly to them. Because in general, the bacteria that are able to grow on MTBE, they're kind of slow growers. They're not always able to, how to say it, to keep them in the bioreactor. But in this case, it's a special material, so it's just a matter of creating the right conditions and getting the right, how to say it, settings for the bioreactor, so we don't have to add any bacteria or biomass afterwards, only one time basically.
Okay, just one last question from Antonio. Do you think that in general, this approach with an external bioreactor that you apply for aerobic degradation can be also applicable for anaerobic degradation?
Yeah, so I mean just the biobarrier concept in principle, if I understand it correctly, that can be used in both cases. We also have applied that, so then it's more like the project Michaela illustrated, the Spanish one. So basically you just have extraction and infiltration wells, but then you put them in a line. So basically you have a biobarrier, you create a biological active zone by dosing your electron donor. So that's basically kind of similar to the aerobic one. I think, yeah, there's no aeration.
To maintain anaerobic conditions. So if he refers to the first project, to use an external bioreactor to the anaerobic system might be possible but challenging, because you have to keep anaerobic conditions in situ. So there might be the risk of air infiltration. So we actually promote the circulation system because it's a closed system where the water acts as a vector to distribute our electron donor and nutrients. But then the system is closed to avoid any air infiltration. I hope that answers the question.
He says many thanks. And I'm going to say as well, many thanks to you guys, to Michaela and John. I think we've run out completely out of time. And thank you very much for such an interesting topic today. And hopefully we will invite you guys to give us an update, maybe in how these projects are going in the future. And thanks everyone, thanks for your time, thanks for dialing in for this webinar. Thanks a lot. Have a great day.
Thank you.
Thank you.
Thank you.
Bye.
Bye.
Thanks, bye.